All right. Thank you very much. I'm going to go a little quick because I got a lot to say. All right. So I'm going to start just by introducing domestication syndrome in case you're unfamiliar with this. So in domesticated species, we often observe that there's going to be this sort of suite of traits that are in common for that species that all domesticated species, at least of a similar type, tend to display. I'm going to be focusing on cereal grains right now, things like wheat, barley, and rye. And the types of traits that are in the domestication syndrome for these are things like a non-shattering seed head, loss of seed dispersal mechanisms, reduced dormancy, and then also increased size. There's a couple others as well, but the one I want to focus on here is the idea of a non-shattering seed head. So you can see in my AI-generated image of wild grass that the seeds here are smaller. And if you've ever been out playing in the wild grass, you can kind of pull them off really easy and throw them off. This is what we call a shattering seed head. This is beneficial for the grains because it allows them to spread their seeds when they're ready. But when we consider domesticated wheat, the seed heads are much more firm. You cannot separate the seeds from the head. And this is what we will call a non-shattering seed head. And anthropologists tend to use this as a signifying feature of what a domesticated grain is. So when they're looking at the archaeological record and they're looking at grains, if they're shattering, then they're going to assume they're wild. And if they're non-shattering, they're going to assume that they're domesticated. So there's sort of two extreme hypotheses related to domestication syndrome. I call them extreme because there's a bunch in the middle in the spectrum that are probably more likely. The first one here is that domestication syndrome arises due to direct, deliberate, artificial selection. So humans, ancient humans were like, we want non-shattering seed heads. So we're going to go find the ones that are non-shattering. We're going to deliberately plant those ones. It kind of puts a little bit of genetic knowledge into our ancient peers that maybe we don't want to attribute to them necessarily. But this was sort of a default hypothesis at the beginning of domestication theory that it must have been us because look at how great we are, of course. Well, recently there's been a shifting towards a different perspective, which is that really a lot of these traits may have been due to indirect natural selection. So really humans just are kind of like predators in this case. They've just gone ahead and interfered with the life cycle of this species. And as a result, they've introduced natural selection pressures that may have caused domestication syndrome. So the research question that I tried to solve with my peer was, well, what role does direct artificial selection play in domestication syndrome? So to do this, we've created a simple simulation. We've got a cellular automata, 50 by 50 grid here. There's some environmental variability. You can see there's a river down the middle that determines water level for that particular cell, and it sort of drops off as you go to the dry region. This determines a little bit about the growth rate of our wheat as well as how many seeds they produce. It was just intended to create a little variability. We also notice on the side here, there are some shelters. And this is just really a granary for the humans. When they gather the grains, they go and put them in this big shared granary. And then later on, they might use that to determine what they might want to plant. We're mostly focused on the genetics of our grain species. So we've got a very simple life cycle built into our grain species. The seeds at first might take root in the environment, then they're going to grow over some period of time until they reach maturity. When they reach maturity, new seeds will appear on their head. It's at this point where humans might be able to harvest those seeds. And then also after a bit of time, depending on their genetics, they're going to drop their seeds and then repeat the cycle. So we've created 4 real value genes that control this growth and reproduction cycle. I'm going to start with the one in the top right here, root depth. This one is relative to the environment. This one, there's sort of 2 variables here. All my real value are from 0 to 1, and I'm kind of just describing their extremes here. But in when you have what we call shallow roots, then the water level of the environment completely determines your growth rate. But if you have deep roots, then your growth rate is just fixed regardless of where you are in the environment. We've also selected the fixed growth rate to be the average growth rate of the environment, so that in the wet environment you'd rather have your shallow roots, but in the dry environment you'd rather have deep roots. Our fecundity gene helps determine how many seeds are produced on the mature plant. We introduced this one mostly just to have we thought there would be a strong selection pressure, natural selection pressure to produce as many seeds as possible. And we were just curious what would happen when we try and apply artificial selection pressures to this gene. The abscission gene is probably our most important one. Abscission is the process by which a plant might separate part of it off, like dropping a leaf or dropping a seed. And so this gene controls when the mature plant is going to drop its seeds to the ground. And we have two extremes. Either we try and immediately drop them, or we're going to delay for some period and then we're going to drop them. The last one, our dispersal gene, it has to do with where the seed drops in the environment. So we just have a simple one. Either we're going to drop into the current cell or we're going to drop into one of our 8 neighboring cells. And this is just a random choice. So depending on what your gene is, we flip a coin and then we pick one of those two scenarios. Our humans are, in this case, they're just homogeneous sort of robots. They all do the exact same thing. They're intended to, we sort of vary their strategies in our experimental settings. So they do two things for us. They harvest, so that means they're going to go out into the environment and pluck seeds. We have 2 settings for that. In our control setting, there's no harvesting, there's no humans at all. We just want to see how natural selection impacts our grains. But in our experimental setting, our agents are going to do random harvesting. So they're going to just pick a random seed in their more neighborhood, step into that cell and pluck it. We considered other strategies, but we're going to push that to future work. Our planting strategy, planting is just a little bit more complex. So I mentioned they've got this big granary where they store all the grains that they harvest. They eat from that. We usually call that the food stores, but then they also have a planting store, so a separate sort of part of the granary where they store seeds for planting. And we can consider a couple different ways that they might move seeds over to that planting store periodically. First of all, they might do no planting, so that's another control run just to see what happens when they harvest with no planting at all. We've also considered random planting, so no selection when we move them over into the planting store. We just uniformly pick seeds. And then all of our experimental settings for our 4 genes, we can consider optimizing those genes. So this would be their artificial selection imposed by the humans by saying, we want non-shattering or we want shattering or something like that. And they can minimize or maximize either of our 4 genes. Okay, let's take a look at some, well, this is sort of our control settings first. So I want to start with the far left column here. This is wild type 1. Wild type 1 occurs when there's no humans at all. This is just natural selection on our grains. We can see a couple patterns maybe that you would have already considered. So for our root depth, you see there's sort of 2 kinds. There's the wet kind and there's the dry kind, okay. For fecundity, yes, we had strong selection pressure to maximize. For our seed dispersal, there's sort of a sweet spot where we don't always want to drop into the local one. We don't always want to drop into our neighbors. We want to have some blend. And so they've sort of evolved to a nice sweet spot. And then for our abscission gene, at least in the absence of humans, there's actually no reason for them to try and shed their seeds as soon as possible. And there is a small selection pressure built into our system down to 0 for all of our genes. So what we're just seeing here is that weak selection pressure is just pushing it down. I think what's really cool is when we move to the next column, we've now introduced harvesting, but no planting. So in this case, we can kind of treat the humans as just a predator on the wheat. They're just trying to eat it and they're not redistributing it in any way. In this case, we see the selection pressure on the abscission gene changes. It says now the plants want to avoid getting eaten by the humans because that's taking them out of the reproductive cycle. And so instead they want to shed their seeds as soon as possible. And again, following anthropologists, this is usually a signal these are wild seeds, okay? So this is going to be the beginning of our wild type. The last one here is just when we also do random harvesting, so random planting, non-selective planting. So we'll just pick any seeds from our planting and we'll go, from our granary and we'll plant them. What we'll see here is they still kind of look like wild type seeds. They still have the shattering type, okay? There is a collapse of our dry type, but if you have questions about that, I'll answer them later. The important thing here is we're going to still consider wild type 3 as a wild type. And we're doing this partly for ease because it's just an easy thing to do, but also to follow sort of the anthropologists that we're looking at. This is how they would divide up the seeds. So we would call this one a wild type. And so from now on, we're just going to use this as a definition. This is our definition of wild type, those that have shattering seeds, and then domesticated type will have non-shattering seeds. So now let's consider one of our experimental runs where what we're trying to do is the humans are going to plant seeds that are non-shattering. So that's sort of our hypothesis one. Let's assume that they deliberately said, we want non-shattering seeds, so we're going to go out and plant them. This is the combined population. And what you'll notice is it looked like, at least to us, that there were two different populations. So what we did is we went and separated those seeds into two piles, those that looked like they were wild type and those that looked like they were domesticated. And then what we tried to do is, so first we identified that there were two clear populations, but then what we wanted to do is look at the genetic distributions on the other genes. Okay, so these ones were selected for, but what we see on what's here, our seed dispersal gene, is that we have a different genetic distribution between the wild type and the domesticated type. This is what we're considering to be domestication syndrome in this particular gene. We can also see why this might be true, that this is the seed dispersal. This determines where they drop naturally, if they were dropped naturally by the plant. But what's happened is humans have gone in and interrupted this part of their life cycle. They take them out and then they go and deliberately plant them somewhere else. So there's no benefit to the organism to have some special dispersal mechanism. And then that weak selection pressure that just says, okay, get rid of those mechanisms because they were costly for us, pushes it down. We also see a change in the root depth. Again, I might talk more about that in the question section if you have questions. The only thing we do notice is there doesn't seem to be domestication syndrome on the fecundity gene. I say there doesn't seem to be, but in some of our runs when we select for that, we do see a small amount of domestication syndrome as well. Okay, so what are some of the conclusions we can draw away from this? One, there was some direct selection involved to get our abscission gene here, but then indirect selection contributes to the others. Let's look at another example. I don't have time to go through all of 8 of ours, but this is just another one where instead of selecting for that abscission gene, we're now selecting for the root depth gene. And I've decided to pick one where we minimize this because that's consistent with what the wild type does. So now we have an artificial selection pressure going in the same direction as the natural selection pressure. As a result, the domesticated variant is much weaker. We can kind of see this line here represents about 10% of the population, whereas previously our domesticated variant was the dominant variant. So in this case, the wild type is dominant and the domesticated variant struggles to sort of keep up. So we, from our results, we decided, or we came up with sort of 4 observations or conclusions. The first one sort of confirms what is kind of the popular belief in the domestication literature right now, which is indirect selection is sort of a major important component to describe or to explaining domestication syndrome. The second conclusion is the one that I think is a little bit more surprising. In our simulation, we could not get any domesticated variant or domestication syndrome unless there was some direct selection on one of the traits. This is meaning that at least artificial selection has to be in there somewhere for us to get domestication to occur. I think this one's a bit surprising and worth testing in other scenarios just to make sure. And then we have sort of 2 secondary conclusions here. One is that when we are selecting for traits, when our artificial selection pressure is going in the opposite direction of our natural selection pressure, then we can get a nice strong domesticated variant. But if we're going in the same direction, we're going to get a weak one. And in some cases, actually a non-existent one altogether. So one of our runs, we selected for the shattering, that's the wild type, and there was no domesticated variant at all in that case. I think I'm pretty close to end, so I'll skip over my future questions and instead kick it out to you and answer your questions. So, I have 1 minute. Okay, perfect. Then, okay, so in the literature, there's also this hypothesis floating around that says maybe the reason why shattering seed heads are so popular is we were harvesting with a sickle. Okay, when we harvest with a sickle and then we drag the straw back, the seeds that stuck to the head are the only ones that would come back. All the rest would fall off. There's this hypothesis that maybe as a result of that, that's what led to the seed heads. What this is selective harvesting, not selective planting. We had done some initial runs with this. Selective harvesting does not result in domestication syndrome. It's because it's very similar to normal predation. If you are always hunting the biggest you usually get smaller, not bigger. Okay, and so we want to retest this one. We also want to expand this to include a couple other gene types. And then really this whole idea came when I wanted to do this with animals. And I started with wheat, and then I said I got stuck on wheat. So I do want to extend this to animals in the future as well. Okay, now we'll kick it out to your questions.